Yeah. So in our previous lectures, we talked about what is medicinal chemistry, what is pharmaceutical chemistry, what is drug discovery, the earlier ways and the newer ways of drug discovery. Okay, that is the forward pharmacology and reverse pharmacology, respectively. And then we started talking about the economics of drug discovery, and then we ended that part as well. Now we'll talk about, we'll become more and more sharper. We'll move towards what is drug design, right? For that, we need to understand what are drugs. Okay, what are drugs? What do we understand by the term drug? Yeah. So drugs are compounds which are used as medicines. Most often they are organic, which are often divided into the broader classes. So most of the times, more than 95% of the times, the drugs that we use would be organic. Okay. And less than 95% of the times we'll have inorganic compounds being used as drugs. So we have few of those organometallic compounds, we have few of those antacids, for example, which are inorganic compounds, which do not contain any carbon, right? So carbon containing compounds are organic compounds. Inorganic compounds are the less than 5% drugs which are used as, or less than 5% compounds which are used as drugs. And if we talk of organic compounds or the drugs, they are often divided into broad classes. The three broad classes, namely small organic compounds like atorvastatin, clopidogrel, etc. Then we have biologics. Biologics is a newer class of drugs that has come up. Okay, so here we have infliximab, erythropoietin, insulin glargine. So the insulin glargine is a type of insulin, biotechnologically derived product. This is also a biotechnologically derived product. This is a monoclonal antibody, infliximab, which is again a biotechnologically derived product. So biologics are nothing but biotechnologically derived products, which are most often medicinal preparations of proteins. So proteins can be either natural proteins or recombinant proteins like antibodies. We can have hormones as well. So those all come into the heading of biologics. Okay. Then inorganic and organometallic compounds. So I was just talking about that the organic compounds are more than 95% of the current drug regimen. And biologics and inorganic compounds are less than 5% of the drug regimen that is used in modern day drug arsenal. Okay, so we have inorganic and organometallic compounds which are useful as drugs. Example, we have lithium, platinum based agents such as lithium carbonate, cisplatin, etc., as well as inorganic metals like gallium. So most commonly, drugs are organic small molecules produced through chemical synthesis. And drugs may also be biopolymer based drugs, which are known as biopharmaceuticals. Right? And these are produced through biological processes. And these are increasingly becoming more and more common. So biopharmaceuticals, biologicals, these are all increasingly becoming common. Whereas inorganic and organometallic and organic compounds, these are the trivially known compounds. Okay. Then, in addition, mRNA-based gene silencing technologies may have therapeutic applications, and many mRNA-based gene silencing technologies are being tried out. Okay, they are being tried out so that therapy, the therapy for certain drugs, the cure can for certain drugs can be for certain drugs. Sorry, so the therapy for certain diseases can be done using such type of advanced therapies like mRNA based gene silencing. Okay, so now we have just talked about drugs. Now we need to talk about the targets. So we have understood what are drugs. Let us try and understand what are targets. So targets are nothing but naturally existing cellular or molecular structures that are involved in the pathology of interest, wherein the drug in development is meant to act. So that particular biological target which is naturally present inside the body either as a part of physiological processes or as a part of pathological processes, those are the targets. Okay, so those are the targets wherein the drug in development is meant to act. Now, the targets are classified into established targets. Okay, established targets are precisely those targets for which the understanding is there, knowledge, data about that target is there. So there is a good scientific understanding supported by lengthy publication history of how the drug functions in normal physiology, how it functions in human pathology, etc. Okay, and newer targets are those targets that are not established targets but which have been or are the subject of drug discovery efforts. So established targets are those targets जहाँ पे टारगेट के बारे में, रिसेप्टर, एंजाइम, व्हाटएवर इज द टारगेट, उसके बारे में सब कुछ पता होता है. Whereas new targets are those targets जहाँ पे not much of data is available for the drug discovery to set in. Okay, but still they are being used for drug discovery. So those would be the newer targets. So the majority of targets selected for drug discovery efforts are proteins and enzyme, and most of the times they are GPCR, that is G protein coupled receptors, or they may be protein kinases. So we know protein kinases most of the times for anti-cancer activity. Nowadays we have so many protein kinase inhibitors that are there in the market, right? So those are examples of targets. In an estimate that was done in 2011, approximately 12 years back, 435 human genome products were identified as therapeutic drug targets or for FDA approved drugs. Okay, so more than 400, almost like 500 के आसपास, 500 से थोड़ा कम genome products were identified as to be therapeutic drug targets for drug discovery. So we often say that certain diseases they cannot be treated. Yeah, finding cure for them is difficult, but that's not the case because now we have the human genome project which is complete and we know all those different proteins that are present in human body. So we can try and find out what are their roles inside the body in normal physiology or disease pathology, and accordingly they can be targeted with respect to with the aim of having drug discovery. Now we have talked of drugs, we have talked of targets. Now let us talk of druggable targets. Okay, let us talk of druggable targets or drug targets. So druggable targets are nothing but those targets जिनके against drugs will act. Okay, so see, for any target to be druggable, the drug has to first go and bind to the receptor. Okay, the drug has to first go and bind to the receptor. Okay, and only when the drug binds to the receptor and it is that receptor is eliciting some response, only then we'll be calling it as a druggable target. Okay, there are so many drug targets, there are so many, where anytime we take a drug we always have some of the other side effects, right? So it is similar to that. So any time we have a target which can be bound to by the drug and some response is generated for that drug, then we call for that particular drug when it binds to the target, then we call that as a druggable target or drug target. So definition wise, it is a biomolecular target, most commonly protein or nucleic acid, and it is a key molecule involved in a particular metabolic or signaling pathway. Okay, and that is to be associated with a specific disease condition or pathology or is important in the infectivity or survival of microbial pathogen. So that is what is a druggable target, any biological target that is amenable to drug discovery and drug design. Potential drug targets may be disease causing or they may be disease modifying. So whenever we have to design drugs, we have to first think of the target. Okay, and once we think of the target, we need to understand as to whether the target is going to be disease modifying or it is going to be uh, you know disease causing. Okay, so if we have a disease causing target, we can just inhibit that target. If we have a disease modifying target, then we can just think of either inhibiting or maybe enhancing the role of the drugs. Okay, so drugs will be designed to enhance or inhibit the target function in these potential drug targets. And small molecules will be designed in such a way that they are complementary to the binding site. Okay, now understand this thing. We are moving more and more towards our actual topic, that is computer aided drug design. All right, so we were just trying to kind of clarify our basics, and as we move ahead, we are going more and more towards computer aided drug design. That is what we are here to study. Okay, so small molecules will be designed in such a way that they are complementary to the binding site of the target. So, for example, we have receptor agonist, we have antagonist, we have inverse agonist, or we have the receptor modulators. Then we have enzyme activators or inhibitors, and we also have ion channel openers or ion channel blockers. Okay, so this is the way small molecules are going to bind to the target. And how are the small molecules designed? They are designed in such a way that they have to bind to the active site. For example, if this is the active site, okay, if this is the active site, and the molecule or the ligand or the drug has to go and bind somewhere here, okay, in this part of the active site, then how should be the structure of the drug? The structure of the drug should be such that it should be complementary in the shape, right? तो drug अगर इस shape का रहेगा तब जाके it is going to bind to the receptor का active site. So that is called as shape complementarity. Okay. And the other type of complementarity is charge complementarity. So if the receptor has, say, suppose positive charge in this part of the receptor and it has a negative charge on this part of the receptor. So how should we design drugs? We should design drugs in such a way that they are, first of all, they should be shape complementarity. They should have shape complementarity so that they can go and fit into the active site. Second is it should have charge complementarity. यानी कि अगर यहाँ पे पॉजिटिव चार्ज है रीजन है इन दैट रिसेप्टर सो वी कैन हैव समथिंग ऑफ द सॉर्ट ऑफ फॉर एग्जांपल CO minus and carboxylic acid which if we can put up CO minus in this part of the molecule, then when it binds to the receptor it is going to have ionic interactions. Similarly, if we have positive charge on this part of the ligand, then we will have you know the charge complementarity, which will help in binding interaction of the drug to the receptor. The small molecule should be designed so as to not affect any other off target, since the drug interactions with off targets may lead to undesirable side effects. Further, due to similarities in the binding sites, closely related targets identified through sequence homology have the highest chance of cross reactivity and hence highest side effect potential. So, for instance, if the drug, if the drug has a small molecule, it has affinity not just for the receptor with which it was supposed to bind, but with other receptors which are present throughout the body. Then what will happen? That drug will tend to lose out efficacy. क्यों? क्योंकि that part of the body जहाँ पे receptors है वहाँ जहाँ पे जाके drug को act करना चाहिए था, that is not happening because the drug is binding to all targets in the body, right? So the efficacy will decrease. Point number one. Point number two, because the drug is binding to the other receptors which are present throughout the body, that drug will also give us side effects. Okay, so that drug will also give us side effects. Okay, so that's the reason why small molecules have to be complementary in terms of their charges and in terms of their shapes. Okay, and what kinds of compound go on to become drugs? In order to seek or discover drugs, it is essential to have a good idea of what kind of molecules are likely to become successful drugs before beginning with the process of drug design and drug discovery. Okay, so the normally preferred means of administration of medicaments is oral, whereas there are many exceptions. The majority of orally effective drugs, they obey the Lipinski's rule of five. Okay, so the drugs are going to be effective via the Lipinski's rule of five. They'll tend to follow Lipinski's rule of five, although it is not a rule of thumb, although it is not a nature's law, but still Lipinski and his colleagues from Pfizer They studied more than 500, something near to 1000, 5000 drugs, and they went on to, you know, find out correlations between the drugs which are orally bioavailable and certain properties. And the correlations that they propose to the entire world are called as Lipinski's rule of five. Okay, they're called as Lipinski's rule of five. So the data upon which this rule rest is drawn from 2500 entries. Okay, sometime back I said I think 5000, right? So that was erroneous. Something like 2500 drug molecules were extracted from the USAN, in a US adopted names, the World Drug List and Internal Pfizer Compounds Collection by Lipinski et al. यानी कि Lipinski and his colleagues, and then they proposed these four criteria for orally bioavailable drugs. Okay, so the substance which needs to be orally bioavailable, it should have a molecular weight of 500 or less. तो सबसे पहला criteria क्या है? 500 का molecular weight or less. Okay, then it should have fewer than 5 hydrogen bonds donating functions. So hydrogen bond donating functional groups should be 5 or less. Then it should have fewer than 10 hydrogen bond accepting functions. So hydrogen bond acceptor group should be 10 or less. And then the substance should have a calculated log P between approximately -1 to +5. So if we see carefully, in the first postulate we are talking of 500, second postulate we are talking of 5, third postulate we are talking of 10, and fourth postulate we are talking of +5. So all of these are multiples of 5, right? All of these are multiples of 5, and hence Lipinski in his rules, what he stated was, Lipinski's rule of 5. Now they are called as Lipinski's rule of 5 because all of these numbers are divisible by 5. They are all in terms of 5, right? तो इसीलिए they are called as Lipinski's rule of 5. लेकिन number of rules कितने हैं? 4 हैं. There are only 4 rules in Lipinski's rule of 5. Okay, please don't get confused with this. All right, so in short, for a compound to be orally active, it should have a comparatively low molecular weight. It should be relatively non-polar, and the partition between an aqueous and particular lipid phase is in favor of the liquid phase. But at the same time, possess perceptible and they should possess perceptible water solubility. Because we know if a molecule is excessively lipophilic, then it will not cross the into the cell, or even if it crosses, it will stay inside the cell. If we talk of a lipophilic or rather a hydrophilic molecule as a drug, then it becomes very easy for the drug to be dispersed all throughout the body. Right? So that is perceptible water solubility. यानी कि there has to be a liquid or sorry, hydrophobic lipophilic balance. Okay, there has to be something called as HLB, hydrophilic lipophilic balance. There are many biological active compounds that satisfy these criteria but fail to become drugs. तो इसीलिए हम लोग कह रहे हैं कि Lipinski's rule of 5, although they are a starting point, they are not the rule of law. Okay, they are not nature's rule कि वो follow होना ही चाहिए. Okay, so few of those molecules, they go on to become drugs, others they will fail. But there are comparatively new orally successful active drugs that did not fail to fit into the Lipinski's rule of 5. So if Lipinski's rule of 5 is followed, the molecule may go on to become a drug. If Lipinski's rule of 5 is not followed, then the drug may not become a drug. Okay, that molecule may not become a drug. So that depends. So that's the reason why we are saying it is not a rule of thumb. And this is a helpful guide, but not a law of nature, right? So that is what is talked about of the Lipinski's rule of 5. Okay, and these criteria, they when they are put together, they have accumulated observations and rationalizations in semi-quantitative terms, and these rules embody useful aspects of Hans quantitative structure activity relationship that will be covered later in our topic under computer aided drug design. Okay, under computer aided drug design or molecular modeling. So अभी यहाँ पे we are talking of Lipinski's rule of five, but Lipinski's rule of five के ऊपर से there was another researcher called as Hans who went on to study another set of you know just give me a moment. Yeah, so I was talking about Lipinski's rule of five. So that same idea was used by Hans and his colleague, and what they found out was there are certain physicochemical properties which can be, correlated to the activity, and that is how quantitative structure activity relationship QSAR studies came into the picture. Okay, so that we'll cover later. For absorption and tissue distribution, a drug must be absorbed through successive lipid bilayers before it reaches its target. And drugs may be able to pass through these barriers rapidly, enough to allow therapeutic concentrations to build up. As diffusion is logarithmic function of size and shape, comparatively compact molecules of modest molecular weight are more suitable. So, in other words, what we are trying to say here, ki shape should be something like spherical. Size should be as small as possible. So that is what we have kind of talked about in the Lipinski's rule of five, right? Then, additionally, there must be sufficient water solubility for dissolution and transport to take place. So that means water solubility is also important. So this correlates reasonably with the capacity to donate and accept a moderate number of hydrogen bonds. So hydrogen bond donation and hydrogen bond acceptance. These are two different criteria that are also dealt with under Lipinski's rule of five. There must also be sufficient lipid solubility to allow the drug to enter and pass out of the lipid environment. So जो cell barriers होते हैं, जो cell membranes होते हैं, these are usually lipophilic. Okay, so they should be sufficient. The molecule should have, the drug should have sufficient lipophilicity so that it enters into and moves out of the lipid environment of the cell membrane. So that lipid environment we have talked about in terms of log P under Lipinski's rule of five. Okay, so it is also apparent that the pKa of the molecule in question is also critical because it reflects the polarity of the substance as a function of the pH of the environment. So log P is a very simple term. Okay, it is just partition coefficient. But if we have to talk about the partition in terms of the pH of the surrounding of that molecule of the drug, then we have to talk about this term pKa. pKa is nothing but the acid dissociation constant. Okay, pKa is nothing but the acid dissociation constant. Okay, and uh, we all know that lower the value of the pKa, more acidic the molecule is. Higher the value of the pKa for the given molecule, the molecule will tend to be lesser acidic. Okay, lesser acidic, in other words, correlates to being more basic. Okay, but if since we are talking of pKa, that is acid dissociation constant, we should be talking only about the acidity. If we were talking about pKb, base dissociation constant, then again the same thing applies. Lower the pKb, stronger is the base. Higher the pKb, weaker would be the base, right? So these are interrelated terms, but pKa is the term that is more widely used. Okay, so acids, bases, and amphoteric molecules have strong polarities, hence partitioning behavior that are strongly influenced by this. तो अगर acidic molecule है, basic molecule है, या amphoteric molecule है, then they will tend to have kind of ionization, okay, and that will influence their partitioning behavior, यानी कि log P. तो अगर log P का effect with respect to pH of the environment देखना है, तो we have to talk about pKa. And then the more salt like they become, the poorer is their ability to be absorbed through the lipid bilayers. Okay, and likewise, if they are too non-polar, they will be dissolved in the lipid bilayers and they will remain in there without getting absorbed or without having their effect, without being biodistributed throughout the body. Okay, so that is how Lipinski's rule of five they hold true even in today's date. Okay. Now this is just a pictorial representation of the different ways in which the molecules or the drugs can permeate across the cell barriers or the membrane barriers. Okay. So let's have a quick look into this particular figure and try and understand the cellular uptake and passage of the drugs. Okay. So this is route number one, route number two, route number three, route number four, and route number five. Okay. So once a drug is taken into the body, it may be absorbed. It may be taken up by the cell via the following ways, five different ways. The first way is passage through the porins. Okay. So there are small pores in the cell membrane through which the molecules can pass through. Okay. So for instance, if we have a hydrophilic molecule, so that such molecules and ions also, we have so many ions that are associated with the cells, right? So sodium ions, chloride ions, calcium ions, etc., etc. So all of those ions and those drugs which are hydrophilic can enter into the cell via the porins. So that is route number one. Route number two is active uptake or efflux. Okay, active uptake or efflux. Okay, so this is nothing but the pumps that are present on the cell membrane. So one pump that we have studied is the H plus K plus ATPase, which is also called as the proton pump. ओके तो एंटासिड्स जब पढ़ा था तभी एंटासिड्स में वी स्टडीड अबाउट H plus K plus ATPase inhibitors यानी proton pump inhibitors, right? What is the role of proton pump? To efflux the H plus. Side by side, Cl minus will also be effluxed by passive mechanism and that will lead to production of HCl, right? तो वैसे ही there are pumps that are situated on the cell membrane which can either throw the drug out of the cell, which are called as efflux pumps, or they can throw the drug into the cell. They are called as influx pumps. Okay. Then the third way is passive diffusion into the cytoplasm. This is the most widely used way for the by the drugs to enter into the cells. Passive diffusion happens by itself. So if there is higher concentration of the drug on the outer side of the cell, then by passive diffusion, it will be diffusing into the cell, into the interior of the cell via the cell membrane. Okay, so that is just concentration gradient based diffusion or passive diffusion. Then we have passive diffusion through the cells. So what can happen is, the passive diffusion can help the drug to get into the cell, or passive diffusion can help the drug to pass through the cell barrier and enter into the systemic circulation. Like for example, just imagine that these are two cells that are present at a barrier, like for instance the intestinal barrier, जहाँ पे most of the food and nutrients are absorbed, right? So there, we have those cells which are loosely held. So route number 5 it talks about the pericellular passage. Pericellular यानी ये जो दो cells हैं इनके बीच में जो जो छोटा सा space है उनमें से those drugs would be transported across the barrier. But step number 4 or passage number 4 is what? When the drug is passing through the cell across the cell membrane via passive diffusion and it reaches onto the other side of the cell, यानी कि it is crossing the barrier. Let that be intestinal barrier or blood-brain barrier or any other barrier in the body. Okay, so these are the 5 ways in which the drugs are taken up by the cells or the ways in which the drugs are crossing the membrane barriers inside our body. Okay, so iska explanation jo whatever I have just told now, that is written over here. So if we have smaller water soluble molecules, they will pass out through the pores, yani ki route number 1. Okay, then if we have other agents which are significantly polar and they have to be taken in or thrown out of the cell, then we have those that route number 2. Okay, for example, glucose is absolutely essential for working of a cell, right? Glucose is converted into energy, ATP. So glucose needs to be transported. तो glucose का transporter का नाम क्या है? GLUT transporter, GLUT, glucose transporter, right? So that comes under category number 2. Then we have category number 3, wherein the molecules are partially water soluble and partially lipid soluble, and they will pass in through route number 3, passive diffusion into the cell. Now, route number 4 is the linings of the intestinal tract, okay, wherein the drug will pass through the cell membrane and it will reach onto the other side of the membrane or other side of the cell. And finally, 5 is where there is interstitial junction between the cells, is the cells are loosely held. Okay, intestinal barrier mein, of the intestinal absorption barrier mein, okay. So if we have the cells being loosely held, then we have route number 5 which is followed. Okay, so this is an explanation of how Lipinski's rule of 5 is applicable even in today's world for drug discovery. And this is also an example of how the physicochemical properties of the drug are very, very important in devising, in designing or discovering drugs. Okay, so this is, Lipinski's rule of 5. Yeah. What if the drug is not able to pass through 1, 2, 3, 4, or 5? The last option is give a hypodermic injection. Simple. Just make it into a parenteral formulation and inject the drug into the systemic circulation. That's it. Okay. So injection should be ideally the last resort for formulation. Okay. Injection should be the last resort when we are doing formulation studies. All right. So that is what is the injection. Then, for drugs to become successful parenteral agents administered by hypodermic needle, satisfactory water solubility is overridingly important criteria, and a low to moderate molecular weight is much less important. Okay, तो अगर directly inject करना है drug को into the into the body, then what happens is water solubility becomes an important criteria, molecular weight and lipid solubility, those criteria become less important. Okay, for example, insulin, some glycopeptides, antibiotics such as vancomycin. Okay, so they are directly injected. फिर उनका log p कितना है वगैरह वो सब देखते बैठने की जरूरत नहीं. They have to be much more water soluble. Okay, or at least they have to be administered in the form of salts. Okay, then other routes of administration are transdermal. Then we have sublingual. Then we have pulmonary. Okay, we have vaginal, we have rectal. These are all different routes of administration, and these are less frequently used. And each of them will have features which require adjustment in the molecular properties for optimization. So parenteral चाहिए, parenteral formulation बनाना है, water solubility is most important. If water solubility is not there, then what we can do is go for some other route of administration like oral administration, or we can go for something like sublingual administration. If we want it to be a sustained release kind of action, then it can be transdermal administration. Sublingual is, you know, equal to sublingual is equal to the parenteral route of administration in terms of onset of action. Okay, so if we have to talk of onset of action, drug action, then जो sublingual है, that is equivalent to injectable. So, for instance, if a person is unconscious, कुछ emergency आ गई है, so that person needs to be injected. But if the person is conscious and if there is an emergency, then instead of resorting to the parenteral route, even the sublingual route can be used. Sublingual यानी the part that is present below the tongue, right? तो जो अपना जबान है, जुबान बोलते हैं, उसके नीचे जो अपना जो एरिया है, that is richly supplied with capillaries, blood capillaries. So absorption happens very nicely from this particular area, and that is why sublingual route of administration is almost equal to that of parenteral administration as far as onset of action is concerned. So some of those nitroglycerines, nitro drugs that are useful for treatment of heart attack or anginal attacks are given sublingually instead of giving as an injection. Pulmonary, for example, asthma hai, we know that the lungs are affected in asthma, right? Bronchial constriction happens and the person is not able to breathe. So if we give something via the pulmonary route, then that is a much better, much quicker onset of action as compared to any other route of administration. Injection with diya to via the blood, the drug has to reach the lungs and then it will exert its action. That becomes a longer way of starting with the drug action. So instead of that, we can use the pulmonary way. But each and every way that we think of, there will be different physicochemical properties that we need to get into the picture and optimize. So that's the reason why we have mentioned that each has its special features requiring adjustment of molecular properties for optimization. Okay. Now, next we are going to start with drug design, or which is also referred to as rational drug design or ligand design. Okay, or NCE, that is novel chemical entity design. So drug design, rational drug design, ligand design, or NCE design is the next topic that we are going to deal with, and we'll do it in our next lecture. Okay, so right now we have finished off with the basics. We have finished off with the background that is required to understand drug design or rational drug design or ligand design as it is called, in detail. So in our further lectures, we are going to talk about drug design. All right.